Hello world. So today I'm actually addressing one of the comments I had from one of my YouTube viewers. They wanted to know what I'm currently doing with my scientific research. So after my grad school, after earning my PhD from Emory University, I got a job at the National Institute of Health, specifically the National Cancer Institute. And here, what I do is I use yeast as a model system to track single particles like proteins in live cells using very powerful microscopes. So I'm going to walk you through a typical day where I use these microscopes to analyze these proteins in live yeast. So here I'm showing you one of those microscopes by Iberia, a company in Germany that actually made this machine. It takes up the entire room. And today, one of the technicians, Jesse, came and I was able to actually see that the machine is up and running. And I took some images of actual budding yeast cells so I could show you. And I was lucky enough to capture a yeast that was in the process of budding. And you could see the daughter cell, the mother cell, the big one, and then the daughter cell, which buds off from the mother cell. And so I'll zoom in later and show you some of those really cool images. I feel like the luckiest kid on earth. I am in one of the best research institutes working with the most powerful microscopes, one of which there's only two of, maybe three in the whole world. And so I feel like a kid at a candy store. And I can't wait to come in and get my hands on these machines. After I got trained, it's so exciting to see some of these experiments in live cell imaging. So if you ever hear, single molecule tracking, single particle tracking, this is what I do on a daily basis. It is extremely exciting, and it is something that's really, really cool to me. And I remember reading about single molecule tracking when I was in grad school, and I was extremely impressed and amazed. And I was like, one day I would want to do single molecule tracking. And even during grad school, I worked on Drosophila, the fruit fly, which I talked about in previous videos, the model system we use in science to study biology. I've always wanted to work with yeast. Yeast are awesome. They're like super yeast, you know. So this is just one of the coolest tools I get to use while studying yeast. So MinFlux, let me talk a little bit about MinFlux. MinFlux stands for minimal photon emission fluxes. And this is a machine made by Iberia, and we can track the single molecules inside these cells using two-dimensional or three-dimensional tracking. And so on a typical day, I would grow my yeast cells. There's a whole world into, I could actually spend the whole day about how you can go about growing yeast cells, what media to use, and once they grow to like a happy state, the best condition, you can stain them with different dyes. Like think about laser tag, where you have red, blue, different colors of laser tags. And so we can tag the proteins that we're interested in with different dyes. And so when you go to look at it in the microscope, you can see, oh, here's my yellow guy, here's my green guy, here's my red guy, because I've tagged those particular proteins with a tag that can help me detect them later. It's really, really cool tracking these proteins in live cells. And this is me, the happiest postdoc on the planet, using MinFlux, tracking yeast cells, live yeast cells. And you can get different windows, different field of views, and different channels based on the different dyes you use to track the proteins. And so you can follow them, the little dots that you see here. Let me show you. So cool. The little dots you see here from the mother cell, the big cell, and the daughter cell are single particles. And we can actually track them and follow them and see where they go, how fast they go. And so this is what I do, y'all. MinFlux by Iberia. Awesome. And another microscope we use here for our particular experiment and model system is a machine called HILA. It stands for Highly Inclined and Laminated Optical Sheath. I got trained by this from an awesome biophysicist, Dave Ball. And this microscope uses powerful lasers. There are three, but sometimes there's another one we can add to. And so with these three lasers, you can track in three different colors: red, green, and dark or far red or orange. And when I got trained on this microscope, it's a tiny room. They have three cameras here. Here's a microscope, and this is attached to a computer. I'll show you later. And so when Dave trained me on HYLA, he told me that some of these parts are custom built, which means he actually had to use his biophysicist abilities to actually make some adjustment on the microscopes to help us image live yeast cells in 2 dimension and track the particles that we've tagged, we've color coded to actually see how they're moving. And so when I got trained on HYLA, I mean, I spent hours in there. I can spend at least 5 hours a day, Saturday and Sunday, Monday to Monday, just tracking and imaging and looking at how these particles are moving. And you could do different conditions and see how that affects the motility of your protein of interest. So this is another very exciting microscope. The difference between HYLA and MinFlux is that in HYLA, we cannot do 3D tracking, and we're limited in how fast we can do our tracking, whereas MinFlux is super fast, and you can do 2-dimensional and 3-dimensional, and you can do super resolution imaging on MinFlux. So each one has its pros and cons, but depending on the biological question you're trying to ask or you're trying to answer, you can decide which microscope is actually more suitable for you. So for my particular research interest, tracking single particles in live yeast cells, my two main powerful tools are HYLA and MinFlux. And sometimes I can do experiment on one and the other and combine and see which one is better. Sometimes you can compare and contrast. And it's always exciting when one machine confirms or validates or consolidate what you've acquired in the other one. And so sometimes that consistency is really exciting and gives you a boost of confidence. All right, I'm doing this right. So it's always a good gut check. So Dave, you are awesome. You are a baller. His last name is Ball, so I always call him Baller Dave. We actually had really great time learning this microscope, and now I can confidently say I'm a HYLA imaging expert. And he also trained me on MinFlux, and so did my boss Tatiana. So it's been a huge and exciting collaborative effort. I love it. I love learning. I love trying new things, and I've always wanted to work with yeast. So it's a win-win. It's the best of both worlds. And it's extremely exciting working on machines that no one else is using or has limited access to. So HYLA is connected to a computer, and I can actually use this joystick here to move the stage so I can see which patches of cells I want to image. Sometimes some patches of cell don't look too happy, so you don't want to image those. You look for another patch. They look cool, they look happy, and you get trained and know what a happy yeast cell looks like after well, I've been here a year and a half, so after a while you get a hint, you get a picture of it, and you figure it out. So here I'm showing you a 2D image of two proteins that I've tagged. One is in red, one is in green. So RFP, red fluorescent protein, GFP, green fluorescent protein. And you can see how these proteins interact, if they come close to each other, if sometimes you think they co-localize or they're located in the same spot, or sometimes it can tell you how far apart they are or how close they are, which direction they go. And you can get so much information about single molecule tracking with these microscopes. And from HYLA 2D tracking, you can actually get a distinct subpopulation of motility, where some particles move very slowly, some move not too slow, not too fast, and then others move very fast. So you get to see how they're moving, how fast they're moving, where they're moving, and you get a lot of information about these proteins, motility, which you can then use to infer the biology. If they spend too much time in one area, what are they doing there? What's keeping them there? So those are all questions that you can answer based on the analysis you do from these experiments. And finally, from these custom-built microscopes and the Minflux from Iberia, company in Germany, you can use different model systems. In my case, I'm using budding yeast, Saccharomyces cerevisiae, to ask basic biological questions. What do proteins do in the nucleus of the cell, the center, or the cytoplasm? What do proteins do when their cells are in a toxic environment or they're stressed? How do they behave? So you can ask so many different questions in so many different model organisms, and you get so much information based on how you're actually studying these biological questions. And so we were really, really excited to actually publish a review on the impact of Saccharomyces cerevisiae on the field of single molecule biophysics. So using Hyla, Minflux, and other microscopes I did not have time to talk about, because these are the two babies I work with, you can ask different biological questions, and you can use different microscopes to help you answer those questions. And so this is a comprehensive review that I was able to be a part of. I am an author on this paper. And if you get a chance to look at it, you'll learn so much about why we use yeast for single molecule tracking, how we use yeast for single molecule tracking, and how you can do it if you wanted to, and the things we can ask and get answers from, the way we analyze these data, the way we track these data, the way we collect the data. It's so much information, but it's a review, and so it's supposed to be so much information. And so many people have been doing this for a long, long time. And so using these machines and these model systems, you can actually attack different parts of the central dogma or the molecular molecules in those in those paths. So for example, if you want to study DNA processing, DNA replication, what happens to DNA when there's a mutation or there's a change in the DNA. So there's whole different topics called epigenetic. I can actually make a whole different YouTube video about that. So you can use model system to study DNA. And remember the cookbook analogy I gave you last time? So if DNA is the cookbook, all the information is there, all the genetic material. And so DNA can be replicated, make copies of itself, and it can be transcribed during a process called transcription to make RNA. And in that case, we have a recipe. And you can have different RNAs or different recipes that can be translated to make proteins. So different RNAs can make different proteins. And so I'm studying how proteins are moving inside live cells. So that's why I have the red box on the protein. And remember Mr. Penguin says DNA gets all the glory, but proteins do all the work. They do. Proteins are the workhorses of the cells. And I'm using these cells, which are unicellular, so I don't have to worry about too many different processes going on. So it's really, really easy and cool being able to actually study protein function and motility in these lively cells. And so I hope you liked this video, and I hope it was also a review on the central dogma on biology. And so if you ever want to know what I'm doing, a simple word would be Binter studies single particles of proteins moving inside cells using yeast, budding yeast. So budding yeast is an awesome model system for studying single particles. And so doing single particle studies with proteins and things like that is really, really cool. And there are so many different genetic tools on using the yeast as a model system to study RNA, to study DNA, and to study protein. So you can do it all. Yeah, thanks. I hope you enjoy, and I hope you learned something new today. Have a great day. Thanks, and please feel free to subscribe, ask any questions, and I will be happy to follow up and actually upload more videos on anything that's not clear.